Good morning, students. Good morning, who has joined to our lecture. Today we are having lecture number 8. Firstly, I want to remind you that we will have microbiology credit in the end of December. So, for those students who have problems in microbiology, I mean missed classes or colloquiums that you haven't passed yet, you must rework your classes up to the end of this month, up to the end of December. Today is the 10th of December, so you have about less than 20 days. Sure, we will not take your reworking on the 30th and 30th of December on the 31st of December. You had a lot of time during semester to study microbiology, and you have time now. On the 30th and 31st of December, we will not take your reworking and we will not reply your messages. You don't have time for microbiology during semester, so we will not have time for you before New Year. Most groups, almost all groups will have the last practical class. The theme of our lecture today is pathogens of Escherichiosis and Dysentery. We will talk about causative agents of typhoid fever, gastroenteritis and cholera. In other words, we will talk about pathogens that cause intestinal infections or infections of digestive system. There are some pathogens in this group that cause infections of digestive system. And the first pathogen that we discuss today is Escherichia coli. So, firstly, we are going to talk about biological properties or biological features of Escherichia coli. This pathogen belongs to family Enterobacteriaceae, genus Escherichia, and species is called Escherichia coli. You remember that last practical class, we also talked about some pathogens from this family. These were Klebsiella and Proteus. So today we also will talk about some members from this family. I remind you that Enterobacteriaceae are mainly bacteria that live in intestine, in human intestine and in intestine of animals, of mammals. Let's talk about morphology firstly. Escherichia coli are straight gram-negative rods as other Enterobacteriaceae. They are motile, they are peritrichous, they non-sporing bacteria, don't have spores, and they're capsulated. What about cultural properties? They are facultative anaerobes. They can live in both presence and absence of oxygen. They can grow on simple media like meat, peptone, agar. And sure, we also can use special media that is used for other Enterobacteriaceae. It is endomedium. I remind you that it is differential media for Enterobacteriaceae. And we can see that Escherichia produce metallic sheen of colony surfaces, and these colonies are non-viscose. They are spherical, non-viscose colonies. And color of bacterial colonies is pink on this medium, because I will tell you in 1 minute that this bacteria ferments lactose. Look at this picture, at this diagram. In this diagram, in the first one, in the left one, we can see colonies of Escherichia on endomedium. We can see that they are spherical like most other bacteria. Color of the medium is pink. I remind you that endomedium is colorless when it is sterile, when there are no any colonies of lactose fermenting bacteria. When we inoculate E. coli on the surface of nutrient medium, of endomedium, bacteria ferment lactose into acid. Acid changes pH, and indicator changes color of the medium. And for example, on meat peptone agar, on MPA, you can see that colonies are white. What about fermentative properties that are used in bacterial identification? The first main fermentative feature of Escherichia, its ability to ferment lactose. That's why we use endomedium. This bacteria also ferments glucose and other sugars into acid and gas. Indole test is positive, so these bacteria ferment peptones into indole. The another one, a Voges Proskauer test. It's test for anaerobic fermentation of glucose. It is negative. It's also used in identification. And citrate test is also negative. These are the main identification tests for Escherichia coli. What about antigenic structure? Antigenic structure of Escherichia includes 3 antigens. O antigen, that is lipopolysaccharide of cell wall. And there is one interesting thing for Escherichia coli. There are about 173 variants of O antigen. What does it mean? For example, when scientists studied strains of Escherichia coli isolated from patients in other countries, they understood that all strains, all isolated bacteria of Escherichia coli had the same properties, the same morphology, the same fermentative properties, but they had the only difference in structure of lipopolysaccharide that is present in cell wall. Then it was discovered that there are, and today it is known that there are about 173 different variants of lipopolysaccharide. They are present in different parts of the world, and they cause different types of infection. For example, antigenic or antigen, if we are talking about this one, it is called O1, O2, O3, O4, O5, O6, O7, and so on, up to O173. And we know that early numbers of O antigen, O1, O2, O3, up to 8, for example, are non-pathogenic bacteria, are non-pathogenic Escherichia coli. They live in our intestine as a normal microflora, as members of normal microflora. But late variants like O22, O56, O156 are very pathogenic Escherichia coli. This antigen is thermostable because it's almost not destroyed by high temperature. The another one is H antigen. It is flagellar because Escherichia coli has flagella, it's motile, and there are about 75 variants of H antigens. They are also called H1, H2, H3, and so on, up to H75. And the last one is a K antigen. It is capsular, and there are about 103 variants of K antigens. Or the same. The rule of these antigens is the same. We call them K1, K2, K3, and so on. K antigen can be thermostable or thermolabile. Some strains have capsular antigen that belongs to group A, that is thermostable, and some strains of Escherichia coli have K antigen that belongs to group B, that is thermolabile, is destroyed by high temperature. H antigen is always thermolabile because it is protein. Flagella of bacteria is made of protein. That's why it is destroyed by boiling, if we boil bacteria. Virulence factors. I remind you that virulence factors take part in pathogenesis. They take part in, they help in development of infection process. The first one is capsule. You remember that capsule of all bacteria have the same main function, important function. What is this function? It is adherence. Firstly, adherence. It is not written here, but you must remember. And the another one, that capsule has antiphagocytic action. It inhibits phagocytosis. The another virulence factor is O antigen. We have said about it. It also protects from phagocytosis. Then fimbria. These are hair-like structures that cover over the surface. They are shorter than flagella. Fimbria are also necessary for adherence to mucosal membrane. Adhesins. Adhesins are specific proteins of the bacterial surface that also help to adhere to mucosal membrane. Then, what about toxins? Escherichia coli produce some types of toxins. The first one is endotoxin. This bacteria is gram-negative. You remember that all gram-negative bacteria always produce endotoxin when bacteria is destroyed. Endotoxin causes fever in human organism. And the another one is enterotoxin. I remind you that enterotoxin is a type of exotoxin. So when you fill your table for bacteria, You may write plus in both columns. Endotoxin is produced, exotoxin is also produced, because enterotoxin is a type of exotoxin. Enterotoxin causes increased secretion of fluids in human intestine and cause the main clinical manifestation of diseases caused by Escherichia, that is diarrhea. There are two types of enterotoxins. The first one is heat labile and the other one is heat stable. They are produced, heat labile and heat stable, they are produced by different strains of Escherichia. And the last one is hemolysin. Hemolysin is an enzyme that destroys red blood cells. It is mostly produced by strains of Escherichia that cause infections, such infections as meningitis, pneumonia, for example. Now let's talk about epidemiology and pathogenesis of diseases caused by Escherichia coli. The first one that we should say here is about the source. The source can be sick man with clinical symptoms or bacterial carrier. You must remember that most infections caused by Escherichia are endogenous. You know what is it? Endogenous and exogenous infections. Exogenous infections, when bacteria enters from outside. Endogenous are infections caused by bacteria of normal microflora. Also, strains causing gastroenteritis are generally acquired exogenously. What are transmission ways? All infections of digestive system have the same transmission ways. It can be direct contact in touching when hands are contaminated with bacteria after toilet, for example, or in Escherichia it can be the another case when medical staff has contaminated hands also, and bacteria can be transmitted in medical manipulations, and it causes hospital infections. You remember what is it. And the another one that is most known is fecal oral transmission via contaminated water or food. All diseases that are caused by Escherichia coli can be divided into two groups. The first one includes extraintestinal infections, and the second one includes intestinal infections that can be called by the word gastroenteritis. The first one, that is extraintestinal infections, look here. We can understand that these infections don't occur in intestine. They occur in other human organs and sites. Extraintestinal infections are mostly hospital infections. They are transmitted from medical staff through medical manipulations. So what are these infections? These are urinary tract infections, then neonatal meningitis, and sepsis. Sepsis develops when normal host defenses are inadequate, destroyed, and Escherichia coli may reach the bloodstream. What about intestinal infections? Intestinal infections are caused by pathogenic E. coli that enter human organism from outside. Let's come back to extraintestinal infections once more. Extraintestinal infections are mostly caused by Escherichia coli, that is non-pathogenic, that live in human intestine. But when Escherichia coli leave intestine and enter other human organs, that's why their Escherichia coli may cause infections, like in urinary tract or in meninges or in blood. Intestinal infections are caused by pathogenic E. coli. So there are two groups of E. coli that are non-pathogenic, that live in intestine, that is normal microflora, and pathogenic that enter from outside. The first one, that is non-pathogenic E. coli, we can call it opportunistic E. coli. Opportunistic, you remember, these are bacteria that cause infections in special conditions. In special conditions, I've said, when bacteria enter in other organs or sites, or in weak immunity. Now let's talk about groups of intestinal infections that are caused by pathogenic E. coli. There are 5 groups of this bacteria. The first one is enterotoxigenic Escherichia coli, ETEC, its short name. The another one is enteropathogenic Escherichia coli. Then enteroaggregative Escherichia coli. Then enterohemorrhagic Escherichia coli. And the last one is enteroinvasive Escherichia coli. The name of every group arises from important characteristic of bacteria or important ability of bacteria or some features of its pathogenesis. You will understand what I mean. The first group that is less pathogenic than others Escherichia coli is enteropathogenic. It includes such strains because of O antigen that are O26, O55, O86, O111. Enteropathogenic Escherichia coli infect just small intestine. It is transmitted by person to person. In other words, it's transmitted by contact, direct contact. What about development? What about pathogenesis? Enteropathogenic Escherichia coli attach to epithelial cells of small intestine. Look here, attach. Then it destroy microvillus. It attach to microvillus and then destroy it and form cup-like structure. It means that microvilli become shorter, and fluid is not absorbed in intestine. You know what is main function of microvillus in intestine? They increase square of intestine. That's why they provide absorption of water in intestine of human organism. When enteropathogenic Escherichia coli adhere to the surface of microvilli, they destroy them, make it shorter. That's why there is no absorption of water, and it causes infant diarrhea in tropical countries. Infant diarrhea. It develops in little children from in newborn up to the age of 2-3 years old. And symptoms of infant diarrhea include severe watery diarrhea, vomiting, and fever. Diarrhea is without blood. It's very important because epithelium is not destroyed here. Mucosa is not destroyed and epithelium is not destroyed. So it's just pathogenic. Pathogenic. It doesn't produce any toxins. And we can say that it's milder than other gastrointestinal infection caused by Escherichia coli. The another one is enterotoxigenic Escherichia coli. It includes such strains as O6, O8, O15. It also infects small intestine. This bacteria can be transmitted through fecally contaminated food or water. The previous one is transmitted by contact. This one is transmitted through food. Enterotoxigenic Escherichia coli is called enterotoxigenic because it produces enterotoxin. We know that enterotoxins stimulate hypersecretion of fluids and cause diarrhea. So, the symptoms of gastroenteritis caused by enterotoxigenic Escherichia coli are similar to those of cholera. What are the symptoms? It's watery diarrhea, vomiting, cramps, nausea, and low-grade fever. If we have some patients with diarrhea, nausea and vomiting, it is impossible to understand which one has cholera and which one has gastroenteritis caused by enterotoxigenic Escherichia. We may understand it just after diagnosis, after bacteriological method, for example. Cholera and gastroenteritis caused by enterotoxigenic Escherichia coli are similar. It causes travelers diarrhea, infant diarrhea in developing countries. Travelers diarrhea is a type of infection that occurs in tourists, in persons who travel from developed countries, like European countries, for example, in developing countries. There, the first 3 days, the first 3-4 days, they have diarrhea and vomiting. It is caused by enterotoxigenic E. coli that can be ingested with contaminated food. Because bacteria multiplies there, and these countries, developing countries, are most countries with a wet, warm, hot climate. Enteroaggregative Escherichia coli also infects small intestine. So pay your attention that the first 3 groups, that is enteropathogenic, enterotoxigenic, and enteroaggregative, infect small intestine, because the other 2 groups that I will explain infect large intestine. Enteroaggregative Escherichia coli cause infant diarrhea in developing countries, travelers diarrhea also, and persistent watery diarrhea that lasts more than 2 weeks. They are called enteroaggregative because they have a great virulence factor as adherence factor, for adherence, for attachment. Escherichia coli with high level of adherence, they have adhesins, capsule, very great capsule. These Escherichia attach to mucosal membrane and they form structures like stacked bricks. Look here, these are microvilli. They attach to the surface of small intestine, to mucosa, and they form structures like stacked bricks, so they cover over the surface of mucosa. They cause shortening of microvilli, shortening, and they may cause hemorrhages. Because of shortening of microvilli, fluid absorption appears. Fluid absorption decreases, and it causes watery diarrhea. Water is not able to absorb into mucosal membrane. And the another group that are enterohemorrhagic Escherichia coli includes such strains as O157H7, it is O antigen, it's H antigen, and O26H1 strains. Why have I said about this one? This strain is one of the most dangerous strains that cause gastroenteritis outbreaks in most countries. Enterohemorrhagic Escherichia infect large intestine. This group of Escherichia can be transmitted by two ways. The first one is person to person, like direct contact. And the other one is foodborne, through undercooked meat products, water, milk, or fruit juices, vegetables, and fruits that are dirty, for example, that are dirty, contaminated with bacteria. Gastroenteritis caused by enterohemorrhagic Escherichia are the most common in the warm months in developed countries. Developed countries, pay your attention. We have just said about three groups: enteropathogenic, enterotoxigenic, enteroaggregative, that are mostly known in developing countries. This bacteria, enterohemorrhagic, is also present in developed countries. What about their pathogenesis? Enterohemorrhagic Escherichia attached to mucosa destroy intestinal microvilli, resulting in decreased absorption. Then bacteria produce cytotoxic toxins, which disrupt cells of large intestine. Cells of intestine are destroyed, so we can see bloody diarrhea. Diarrhea with release of blood. It is a so-called hemorrhagic colitis, inflammation of large intestine with bleeding, with hemorrhage. With abdominal cramps, we receive abdominal cramps. Abdominal cramps appears because of destruction of cells of large intestine. We can see little fever, or sometimes there is no any fever at all. And finally, it can progress to hemolytic uremic syndrome. What is it? Hemolytic uremic syndrome includes acute renal failure, that abrupt loss of kidney function, thrombocytopenia, it's abnormally low levels of thrombocytes, and hemolytic anemia, that is loss of red blood cells. From the name of this group, we may understand that the main process in pathogenesis is hemorrhages in intestine or in kidney because of action of this bacteria, because of action of toxin. And the last group of pathogenic Escherichia includes enteroinvasive Escherichia coli, that consists of such strains as O127 24, O136, and O154. This group also infects large intestine as the previous one. But this one is rare in both developed and developing countries. It's more rare than the previous one. Enteroinvasive Escherichia coli produce Shigella-like toxin. Shiga, it is name. It arises from name Shigella. Shigella causes dysentery. It will be the next pathogen that we discuss. Shiga, it is name of doctor who discovered it. So, toxin produced by enteroinvasive Escherichia is resembles toxin of Shigella dysentery. That's why this disease develops as dysentery, resembles dysentery, because of their clinical symptoms. Bacteria produce Shiga-like toxin, invade and destroy the colonic epithelium in large intestine, producing a disease characterized by fever, abdominal cramps, and bloody diarrhea. It can progress to colonic ulceration. And I've said that it's like dysentery. So, from the name of this pathogen, we can understand that the main characteristic of this bacteria is the ability to invade and destroy epithelium in large intestine. That's why then it leads to bloody diarrhea, abdominal cramps, and ulceration of colonic epithelium. We have said about pathogenesis. Now let's talk about microbiological diagnosis. Treatment and prophylaxis of diseases caused by Escherichia coli. Specimen choice depends on the type of infection. If these are intestinal infections, we can collect feces, vomiting, food stuff, samples of washings from objects, of vegetables, fruits, for example. If infections are extraintestinal, so we can collect blood, pus for diagnosis, or if it's meningitis, cerebrospinal fluid. Methods that are used for diagnosis are the same like in other bacteria. The first one is microscopy, that includes preparation of smears and its gram staining. The second one is bacteriological, but there is one important thing that you must memorize here. We can use bacteriological method just with agglutination test, because if we inoculate feces on nutrient medium, E. coli always grow, always produce colonies, because they are members of normal microflora. But agglutination test can allow to understand, allow to identify what is O antigen. It's O1, O2, O3, O4 that are non-pathogenic, or maybe O22 or 55 that are pathogenic. So we use it just together with agglutination test. And serological test for detection of antibodies or for identification of antigens. What is O antigen variant, antigenic variant? It's agglutination test and immunofluorescence test. And we also can use a PCR. Treatment includes using of antibiotics that are sulfonamides, cephalosporins, and aminoglycosides. There are no any vaccines for for treatment for prophylaxis of diseases caused by Escherichia. The another pathogen that we discussed today is Salmonella. Both species like Salmonella and Escherichia were named in honor of doctors who discovered them. Escherichia was named in honor of Dr. Austrian pediatrician Theodor Escherichia. Salmonella was named in honor of Dr. Salmon. It also belongs to family Enterobacteriaceae, genus Salmonella, and there are some species. The first one is Salmonella typhi that causes typhoid fever or typhoid, or the third name is enteric fever. In different textbooks you may find different names. Then Salmonella paratyphi A that causes paratyphoid fever A, Salmonella paratyphi B that causes paratyphoid fever B, and Salmonella Enteritis and Salmonella typhimurium that cause Gastroenteritis. Salmonella are gram-negative rod-shaped bacteria. They are motile, non-sporing, non-capsulated. Sometimes some strains may be capsulated. Most of them non-capsulated. They are facultative anaerobes. They can grow on both simple medium and special medium. Special medium is the same, it's endomedium that is used for cultivation of all Enterobacteriaceae, and Meconic also can be used. And Salmonella produce colorless colonies because this bacteria doesn't ferment lactose. And they can grow on bismuth sulfite medium, it's a differential medium, special medium for Salmonella, and they produce black colonies. In these diagrams you can see Salmonella. On Meconic we can see colorless colonies because bacteria doesn't ferment lactose and doesn't change color of the medium. And here we can see colonies of Salmonella typhi on bismuth sulfite agar, and colonies are black. They ferment glucose, mannitol, and maltose, forming acid and gas, except Salmonella typhi. Lactose test is negative, that's why colonies are colorless. They produce hydrogen sulfide when they split peptones and don't produce indole. They have H antigen, O antigen, and virulent or Y antigen. It's a polysaccharide of the surface of bacteria. Antibodies that are against O antigen can't bind with bacteria and destroy it. So it inhibits action of antibodies, action of immunoglobulins. Salmonella have some virulence factors, for example, fimbria, that are necessary for adherence of bacteria to mucosal membrane. They produce catalase. This enzyme protects bacteria from killing in macrophages, so they inhibit phagocytosis. And bacteria can be survived in macrophages. They are gram-negative, that's why they produce endotoxin. It causes many systemic manifestations. They produce enterotoxin. You know, it inhibits fluid absorption in intestine and cause diarrhea in final result. They have invasin. These are proteins that help in invasion of epithelial cells of intestine. And virulent antigen that is related to virulence by inhibition of phagocytosis and resistance to complement activation. Complement is non-specific protein of immune system, and they produce DNA that destroy DNA of human cells. All these virulence factors take part in pathogenesis. So what about pathogenesis of typhoid fever? The first one, the source is a sick man and carrier. Bacterial carrier have a great role in spreading of typhoid fever. Maybe you heard about such case in medicine as a Typhoid Mary. If you don't know who is she, Typhoid Mary, you may look for some information in textbook or in Google about Typhoid Mary. And we'll talk about it in practical class. Transmission includes fecal-oral transmission through contaminated food or water and direct contact. The important thing that you must memorize that immunity after typhoid fever is long-lived or lifelong. It is survived during all the life. Patient can have typhoid just once alive. What about pathogenesis? Pathogenesis is described here in this scheme. You must remember that typhoid fever in patients develops during some weeks, and we can see specific clinical symptoms in different weeks. Firstly, incubation period of typhoid is about from 7 up to 14 days. In most cases it is 1 week. So in the first week bacteria enter the human organism, attach to mucosa of small intestine, enter pair patches, invade lymphatic cells in small intestine, invade epithelium of small intestine. Pay your attention, it is not written here. I haven't written what happens in the first week, but you must know what happens. Once more, on the first week, bacteria enters the small intestine, attach to mucosal membrane, to pair patches, infect lymphatic cells, invade mucosal cells, invade epithelium, and enter the bloodstream. Up to the end of the first week, there is a bacteremia. Bacteremia, you remember, bacteremia is a circulation of bacteria in the bloodstream. What happens on the second week of disease in patient? In the second week, bacteria spread in reticular endothelial system. What are these? These are cells of immune system, neutrophils, macrophages. Then bacteria enter in internal organs, in parenchymatous organs, such as liver, such as spleen, lungs, kidneys. That's why bacteria cause necrosis in these organs, and they cause such clinical symptoms as splenomegaly, it's enlargement of spleen, and hepatomegaly, it's enlargement of liver. And we can see in patient that his or her abdomen is enlarged. So in the second, up to the end of second week, Salmonella Salmonella are present in internal organs, in such organs as liver, spleen, kidneys, in lungs, so we can see cough in patient because of multiplication of bacteria there. Then on the third week, bacteria come out from spleen and they enter through small intestine once more. Some bacteria are present in gallbladder because salmonella like bile is present there, so they like this substance. It's a very nice nutrient for them and they are present there, but some bacteria enter in small intestine once more from liver and they invade epithelium of small intestine once more. That's why we can see ulceration of the pear patches. Pear patches are invaded several times at the first week and then at the third week. It can cause intestinal bleeding and intestinal ulceration, ulcers in intestine. Up to the end of third week, bacteria are released with feces. On the first week, on the second week, bacteria are absent in the feces. That's why we can't collect feces for diagnosis of typhoid. You must memorize it. Then from the fourth week of disease, we can see colonization of gallbladder when bacteria can persist, can be survived during long time, several months. Patients can be chronic carriers when they release salmonella during 1 year or 2 years with feces because of long presence of salmonella there. And chronic infection may involve the joints, bones, meninges, and other sites and cause pain here. It's chronic stage when bacteria are persist in other internal organs. What are symptoms and signs of typhoid fever? In incubation period, we can't see any clinical manifestations. After incubation period, I have said that in most cases the incubation period is about 7 days. Sure, it may be longer. In most, this is 7 days. After the first week, we can see diarrhea or constipation. Diarrhea because of enterotoxin, action of enterotoxin that is produced in intestine by bacteria. You remember, let's come back, you remember that bacteria, as the second week, bacteria spread in lymphatic system, in macrophages, and they enter in internal organs, parenchymatous organs, liver, spleen, kidneys, lungs. So, let's look what can we see there. We can see fever because of destruction of some bacteria via macrophages. Endotoxin is released and it causes fever. Headache, malaise, anorexia, myalgia, arthralgia, cough, and sore throat. And here from the second week, we can see such clinical symptoms as neuropsychiatric manifestation, that is psychosis and confusion, because of action of toxin, it's inflammation, coryza, that is inflammation of nose, as cough, sore throat, chest pain because of development of bacteria in parenchymatous organs like lungs, nausea, vomiting, and abdominal pain. The another disease that is caused by Salmonella is gastroenteritis. It is caused by Salmonella typhimurium or Salmonella enteritis. Sources here are animals, are domestic animals, farm animals that we use to drink milk or, for example, to eat meat from these animals or birds. Cow, sheep, pigs, hen, geese or ducks. And the only transmission way is fecal oral through contaminated food, eggs, milk, and meat. Pathogenesis of gastroenteritis. Gastroenteritis means inflammation of small intestine. Gastroenteritis includes such stages as attachment to enterocytes of the colon, invasion of mucosa by Salmonella, and local inflammation there because of its invasion. Then they produce enterotoxin that causes diarrhea. Diarrhea then illness usually begins suddenly with diarrhea and vomiting, and sometimes with high fever. For microbiological diagnosis, we should collect some specimens, and the choice of specimen depends on the week of disease in typhoid fever. In the first week, up to the end of the first week, bacteria are present in the bloodstream. They cause bacteremia, so we can collect blood. Up to the end of the first week, bacteria are present in internal organs, liver, spleen, but it is impossible to take material from there. It's very difficult, impossible. That's why we can collect serum. Antibodies are detected, appear up to the end of the second week. On the third week, we know that bacteria start to be released from feces, so we can collect feces for diagnosis, and urine, because bacteria are present in kidney. In gastroenteritis, we can collect feces, food stuff like milk, meat, eggs, and blood for serological tests. Which methods are used for diagnosis? The first one is microscopic, the second one is bacteriological, then serological tests that include agglutination test and tube dilution agglutination test, and PCR. What about spreading of typhoid fever in the world? Typhoid fever is endemic in many developing countries, and primarily in developing countries where sanitary conditions are poor. Typhoid fever is endemic in the Indian subcontinent. Look here. It's endemic in all parts of India, but it includes also countries that are nearest to India, Bangladesh, Pakistan, for example. Then typhoid fever also occurs in Southeast and Far East Asia, in African countries, sure, in the Middle East, in Egypt, Iraq, Iran. So in most countries where climate is wet and warm, and where sanitary conditions are poor. What about prophylaxis of typhoid fever? There are more than 2 types of vaccines that are, these are TAB vaccine, that is killed vaccine, then Y capsular polysaccharide antigen vaccine that consists capsular antigen, virulent antigen, and live attenuated oral vaccine. For gastroenteritis, there are no any vaccines. We can follow to food hygiene and avoid uncooked food. Meat, fresh meat, for example, fresh eggs, For treatment, we can use such antibiotics as chloramphenicol, oxytetracycline, and nitrofuran antibiotics. The another pathogen that we discussed today belongs to family Enterobacteriaceae, to the same family. Genus is Shigella, and there are some species: Shigella dysentery, Shigella flexneri, Shigella boydii, Shigella zanae. It is the causative agent of dysentery. Shigella was named in honor of Japanese doctor Shiga, who discovered it, and all other species like flexneri, Shigella flexneri, boydii, and zanae also were named in honor of doctors Flexner, Boyd, and Zanae. Shigella dysentery is called dysentery because of its name of disease. These are also gram-negative rod-shaped bacteria, non-spore-forming, non-motile, and most strains are capsulated. They are facultative anaerobes. They use simple media for growth and special media like endomedium and McConnell agar. They produce convex, circular, colorless colonies because they don't ferment lactose. They ferment mannitol, except Shigella dysentery doesn't ferment, and non-lactose fermenting bacteria, except for Shigella zanae that can ferment lactose and ferment glucose into acid. They have O antigen and capsular antigen, that is K antigen. They have such virulence factors as a capsule for adherence and avoidance of immune action, adhesins that also take part in attachment. They are gram-negative, so they produce endotoxin that contributes, they take part in irritation of the bowel wall. And they produce exotoxin that includes two types of exotoxin. The first one is Shiga-toxin. Some bacteriologists, some doctors call it verocytotoxin. Vero because there is a tissue cultures that are called vero. This toxin causes destruction of vero cells. Shiga-toxin is an exotoxin that consists of two subunits. The first one binds to host cells, to epithelium of large intestine. The second one disrupts protein synthesis in epithelial cells of intestine, so it leads to destruction of epithelium. Exotoxin is another exotoxin that acts as enterotoxin, induce fluid accumulation, and as well as neurotoxin that damage the blood vessels of CNS. Epidemiology and pathogenesis of dysentery. The source are sick men and bacterial carriers. Transmission ways are fecal-oral transmission through ingestion of contaminated food or water, and contact transmission. Dysentery has short incubation period, about 1-2 days. Shigella attached to mucosal cells in large intestine, invade epithelial cells of large intestine, multiply intracellularly, leading to destruction of the epithelial layer. So it is the first step of pathogenesis in dysentery: attachment to mucosa, invasion of epithelium, and destruction of epithelial cell. Then it leads to microabscesses in the wall of the large intestine that leads to necrosis of the mucous membrane and bleeding. Necrosis is the next stage, and bleeding. Shigella produce Shiga-toxin, inhibiting protein synthesis of host cells that damage intestinal epithelium. Each bowel movement is accompanied by straining and tenesmus. Tenesmus is a rectal spasm when the patient wants to go to the toilet, but he is not able for releasing of feces, because patient just thinks that he wants to go to toilet, but it is impossible because this feeling is caused by movement of walls of intestine. And destruction of epithelium results in lower abdominal pain. Not in stomach, in lower, it's lower. After dysentery, there is short-lived immunity. The another main clinical symptoms, bloody diarrhea. Why is it bloody? Because there are bleedings. There is a great destruction of epithelial layer in large intestine. Microbiological diagnosis, treatment and prophylaxis of dysentery. The choice of specimen includes a collection of fresh stool, blood, mucous flax, and rectal swabs. Methods that are used is microscopy, bacteriological, serological, that are in agglutination tests, immunofluorescence tests, and ELISA, enzyme-linked immunosorbent assay, and PCR, polymerase chain reaction. And the another method that is used, we haven't said about it in previous lectures, it's the first time when we talk about it, it's allergic test or skin allergic test. This method may be done because of exotoxin that is produced by Shigella. Exotoxin is injected intracutaneously and then the result is read in 24 hours by the size of the formed papilla, here or in the forearm. That test is considered markedly in the presence of edema and skin hyperemia, redness. Positive result of the test if diameter of papilla is 35 millimeters or more. It means that patient has dysentery. Moderately positive if this diameter is from 20 up to 34 millimeters. Doubtful if there is no papilla and the diameter of skin hyperemia measures from 10 up to 15 millimeters. And test is negative. We can say that patient has no dysentery if the hyperemic area is less than 10 millimeters. So positive result if diameter is more than 35 millimeters, negative is less than 10 millimeters. What about treatment and prophylaxis of dysentery? Treatment includes using of such antibiotics as ciprofloxacin, ampicillin, doxycycline. For risk group people, we can use chemical vaccine. Risk group people who live in regions with outbreaks of dysentery. And the last pathogen for today, is Vibrio Cholera. Pay your attention that this pathogen belongs to another family, Vibrionaceae. Escherichia, Shigella, and Salmonella belong to Enterobacteriaceae. Vibrio belongs to Vibrionaceae. Its genera called Vibrio because its shape. You remember from the first lecture, Vibrio are comma-shaped bacteria, like a comma or curved rods. And there is the only species in this genera, it's Vibrio Cholera. It's the causative agent of cholera. Vibrio Cholera classic, and the another biological variant is Vibrio Cholera L. Tor. Firstly, let's talk about discovery. Vibrio Cholera was discovered by Robert Koch in India in 1884. Robert Koch was German scientist, German bacteriologist. When there was an epidemic of a new disease in India, when a lot of people died, Robert Koch decided to go to India to find what is the causative agent, why a lot of people died because of this disease. By that time, Robert Koch uh, got married, just married. So it was a honeymoon with his young wife, and they went to India. They looked for pathogens. They looked for pathogen in all rivers, in all reservoirs with water, in sewage. So Vibrio cholera was discovered by this great scientist. After some years, there was outbreak of the same disease like cholera, but this pathogen had little bit other properties. It was called later Vibrio Cholera L-Tor. It was first identified in 1905 at a quarantine camp on the Sinai Peninsula in El-Tor in Egypt by German physician Dr. Felix Gottschlich. The vibrios were found in the guts of 6 pilgrims who returned from Mecca, and they stayed in El-Tor, or Ol-Tor, it may be called. And some of them died, some of them have infections that was resembled cholera. Vibrio cholera classic is distinguished from Vibrio cholera L-Tor. There are some differences in resistance to antibiotics, in hemolysis. For example, Vibrio cholera classic doesn't lyse red blood cells. Vibrio cholera L-Tor cause lysis of red blood cells. Vibrio cholera classic doesn't cause agglutination of red blood cells. The second one, that is Vibrio cholera L-Tor, cause agglutination of red blood cells. Vibrio are have a very interesting shape. They are like coma or curved rods. They are gram-negative, motile. They have single flagella, monotrichous, non-spore-forming and non-capsulated. They are obligate aerobes. That's why if they multiply in water, in river, for example, they multiply on the surface, on the outer layer of water reservoir. They grow in simple media, mid-peptine agar and in peptine water, but they like alkaline medium. They need not neutral. They need pH that is about 8.2. Special media is thiasulfate, citrate, bile, sucrose, agar, and you can see on this image that bacteria Vibrio produce yellow colonies on the surface of this medium. They produce convex, smooth, round colonies. They have O antigen, that is lipopolysaccharide of cell wall, that confer serological specificity. There are at least 206 antigenic groups of Vibrio. You remember, for example, I said about Escherichia. They have about 173 antigenic variants. Here, Vibrio cholera have about 206, but Vibrio cholera strains, so group 1 and group 139 cause classic cholera. Group number 1 was discovered firstly in India. That was the first one that was discovered, and O group 139 was discovered later in Madras in India in 1992. Other strains that are known, O1 known, O190, 39, sorry, Vibrio Cholera cause cholera-like disease, but it's not cholera. Vibrio Cholera L. also belongs to O group 1, and they have H antigen. They coagulate serum and gelatin, breaks down urea, and ferment sugar with acid formations. They produce some virulence factors as exotoxin, that is also called chloreogen, causes hypersecretion of large volumes of water. They are gram-negative, so they produce endotoxin that has a powerful toxic effect. Fibrinolysine is also produced. It helps in releasing of free vibrios from the bound mucosa to the intestinal lumen. They produce hyaluronidase that also take part in spreading. It destroys hyaluronic acid. Mucinase that splits mucous, neuraminidase that causes sialic acid of mucous cells, and protease that destroy antibodies. Epidemiology and pathogenesis of cholera. Firstly, we should say that cholera is undoubtedly the most dramatic of the waterborne diseases. The source of cholera are sick persons and carriers. Transmission includes fecal-oral transmission from by food, water, and contact through contaminated hands. Incubation period is short, from several hours to up to 6 days. There are some stages in pathogenesis of cholera. The first one is called Cholera Enteritis or choleric diarrhea. It lasts about 1-2 days. Sometimes the infectious process terminates in this period and the patient recovers. Patient is convalescence after this stage, but if it develops later, then, it leads to Cholera Gastroenteritis. That is characterized by profuse diarrhea, continuous vomiting, leads to dehydration of the patient's body. This results in lowering of body temperature because of dehydration, decrease in the amount of urine excreted, drastic decrease in the number of mineral and protein substances, and the appearance of convulsions. All these manifestations are caused by severe dehydration. These stools, the feces, resemble rice water and contain, because they contain enormous numbers of turn-off intestinal epithelial cells and cholera vibrios. Its color like white, and because of great number of epithelial cells, destroyed epithelial cells and vibrions there. And the final stage is cholera algid, when the skin becomes wrinkled due to the loss of water. Cyanosis appears. Cyanosis when skin becomes blue and the voice becomes husky. Patient can't have speaking or can't speak, and it's sometimes lost completely. The body temperature falls to 35.5 degrees up to 34 degrees. This, which may occur in as many as 60% of untreated patients, results from severe dehydration. Immunity after cholera is long-lived. In this photo, you can see a patient with cholera algid. When we see decreased skin turgor, wrinkled skin, because of loss of water. In this map, you can see spreading of cholera in the world, and you may understand which countries are infected. This, in most cases, these are countries of Central Africa and East Africa, and some Asian countries like India, Asian countries, other Asian countries. For microbiological diagnosis, we can collect stool, vomit, objects contaminated by patient's stools, water, sewage, rectal swabs. Patient must be transported to the lab immediately because Vibrio can be destroyed and they can die in the presence of acid or in the absence of oxygen. You remember they are obligatory roles. There is rapid diagnosis or express diagnosis that includes immunofluorescence test, reaction of immobilization of Vibrios, and PCR. Usual methods of microbiological diagnosis includes microscopy, dark field microscopy, when we observe Vibrio on the dark background. Bacteriological is the next one. You must remember that specimen firstly must be inoculated in transport medium, Pipton water with alkaline, for example, then in enriched medium, because sometimes it is impossible to find some Vibrio in Pipton water, for example, and Vibrio grow faster than most bacteria during 6 up to 12 hours. Most bacteria live during 24 hours. Serological tests are also used, for example, slight agglutination test and polymerase chain reaction. We can use antibiotics for treatment, for example, tetracycline, amphenicol, and streptomycin. For treatment, we also should use replacement of fluid and electrolytes by oral administration of fluid containing glucose and electrolytes, sodium chloride, for example, sodium chloride or physiological solution. Because you know, all pathogenesis is characterized by great loss of water. Prophylaxis includes using of live attenuated vaccine and killed cholera vaccine. Disinfection in departments for cholera patients and in the focus, it's general prophylaxis. And another way for general prophylaxis is a strict observance of individual hygiene, boiling or proper chlorination of water, decontamination of dishes, hand washing. Today we know that Vibrio cholera can't live in acidic environment. That's why scientists say and doctors say that which patients are the most sensitive for Vibrio cholera? These are those patients whose gastric juice is less acidic. It's if the patient has a normal state of gastric juice, normal acidic state. So most Vibrios are not able pass through gastric juice. And you must memorize that, for example, to be protected from cholera, it's enough to boil water during some minutes, during 5 minutes, it's enough. Some doctors say that some Vibrio started to destroy when water starts to boil. That's why these are general rules for prevention, for protection. Now that's all for this lecture. Thanks for your attention. Please don't forget to start to rework this class, your classes up to the end of December. Thanks for attention once more. Goodbye.